everyone. My name is Daniela Gutierrez-Andrade, and today I'm going to be talking to you about a really cool study that I recently conducted along Dr. Michael Middlebrooks and Dr. Sarah Cuccinello on the range expansion and diet expansion of a Chromodorididae Nudibranch Felimare ruthae. I hope you enjoy it. Our knowledge of the geographic ranges inhabited by nudibranchs has expanded due to recent scientific studies, public records, and the increasing popularity of nudibranch underwater photography. However, we are still lacking information on the specific boundaries of these ranges and on the distribution of some species. Recently, range expansions of many different nudibranchs have also been reported across the globe. The causes underlying these geographic spreads are varied. In some cases, the range expansions have occurred due to natural causes, but climate change and accidental introductions through shipping, transport, or the aquarium trade have also contributed to the movement of slugs. Chromodorididae slugs are oligophagous sponge feeders with very specific diets. Although there are some exceptions, most of them feed on only one or two species of sponges that are often very closely related. They don't only rely on these sponges as a food source, they also use them for the obtention of defensive secondary metabolites that will be stored and used against potential predators. Felimare ruthae is a small chromodorididae slug that inhabits the western Atlantic. It is found at shallow depths in areas with moderate currents as far south as Venezuela and as far north as the Bahamas and east coast of Florida. The nudibranch has also been found in the southern Gulf of Mexico, inhabiting the coastal reefs of the Yucatan Peninsula. It is reported to feed on sponges Decidia janaea and Decidia etheria. That is why we were very surprised when on July 2019 we were notified of the presence of Felimare ruthae on the limestone ledges off of Clearwater, Florida. The slug had never been reported on Florida's west coast, so we had to conduct an initial diving survey of the area known as Smith's D Ledge to confirm its presence. On that dive, we observed Felimare ruthae crawling and feeding on large sponges that did not resemble Decidia janaea or Decidia etheria. We visited the site a second time to conduct a formal survey of the slugs and the sponges on which they were found crawling. Tissue samples of these sponges were collected and preserved in ethanol so that we could later morphologically and molecularly identify them. We extracted the DNA from the tissue samples. After that was done, the 28S rDNA-C region was amplified using PCR. Course sequencing was then conducted, and the BLAST tool was used to be able to find similarities between our extracted DNA and other biological sequences. What we found was fascinating. Of the 35 sponges we surveyed, 12 harbored a total of 16 Felimare ruthae. During the surveys, we also measured the diameter of the sponges, but no relationship was found between sponge size and number of nudibranchs. Both the morphological and molecular analyses showed that the sponges on Smith's D Ledge the Felimare ruthae was consuming and inhabiting on were in genus Spongia. This means that the nudibranch did not only undergo a range expansion, it also underwent a diet switch or a diet expansion. This is truly remarkable. Switching diets is no small feat, because in order to consume a sponge, a Chromodorididae nudibranch has to be able to metabolize all the chemicals in its sponge food, and in many cases, it also has to be able to modify these compounds for its own defense. Spongia and Decidia are in different families and possess different metabolites. A diet expansion implies that a significant process of adaptation had to take place. The genus Felimare seemed to have evolved feeding on Decidia. However, another Felimare species, Felimare tricolor, has also been seen to occasionally feed on Spongia. Our results suggest that either some species can opportunistically feed on other sponges, or that certain populations have successfully modified their diet. So, what's next? We know that Felimare ruthae was still around at the beginning of 2020 on Smith's D Ledge. However, the causes of its range expansion still remain a mystery, and we don't really know how permanent it will be. Furthermore, we would also like to establish if other Felimare ruthae populations are also able to consume Spongia, or if this is only exclusive of a Clearwater population. If you want to know more about this, be on the lookout for the upcoming July issue of the Nautilus, and you will find that study there. Also, don't forget to follow us on Instagram at UTampa Slug Life. There is no life like this slug life. Thank you.